Okay, let's do muscle. This is chapter 10, muscle, and it's basically the last chapter of the semester, although we're going to add a little bit of chapter 11 to muscle. So, muscle system and muscle tissue. Functions of muscle, obviously, movement, stability, control of body openings and passages, and heat production. Okay. Universal characteristics of muscle. Like nerve, a muscle is excitable. And excitability, what does this mean? Tissue is able to generate and conduct action potentials. Okay, so, well, that's part of it right there, conductivity. So you can generate action potential, you can conduct an action potential. Obviously, muscles exhibit contractility. They exert a force when they contract, all but one. Extensibility. They can stretch, and that comes along with elasticity. They're stretching, you can stretch them. And as far as skeletal muscle goes, we're going to look at how much strength they can generate is a function of of how stretched they are. They can be understretched or overstretched. It's just at the right length, right degree of stretchiness, then they generate the most force. It's true for skeletal muscle. Oh, and I should say also, this is a chapter on muscle, and there's three types of muscle: skeletal muscle, cardiac muscle, and smooth muscle. We're going to talk about all three, but mostly we're going to be dealing with skeletal muscle because next semester we're going to have a whole chapter on cardiac muscle and smooth muscle. So we're going to get a little bit more, mostly skeletal muscle, a good bit of smooth, and just a little bit of cardiac because, like I said, next semester we got a whole chapter on. Okay. A muscle anatomy. It's a bundle of bundles of bundles of bundles. Bundle of myofilaments make up a myofibril. Bunch of myofibrils make up a muscle cell, sometimes called a muscle fiber. Bundle of muscle cells make up what's called a fascicle, and a bundle of fascicles make up a whole muscle. I'm going to show you that in a moment on the next slide. And it's connective tissue coverings, and endomysium around each fiber. And these are more like dense irregular connective tissue. A perimysium around each fascicle. Epimysium around the entire muscle. And fascia is outside the muscle and attaches the muscle to other parts of the body and separates muscles from each other. Okay, there is the muscle. And so here, these are myofilaments in here. That makes up, so there's a muscle fiber or muscle cell. And skeletal muscle cells are long cylinders. Each individual one, the diameter is microscopic, but the length could be macroscopic. They extend the entire length of the muscle. So a bunch of myofilaments make up a muscle fiber, a bunch of muscle fiber, and it's surrounded by the endomyosin. A bunch of muscle fibers make up a fascicle, and it's surrounded by the perimysium. A bunch of fascicles makes up a whole muscle covered by the epimysium. And then there's deep fascia that's on the outside that may extend in, that covers the muscle and may extend to the bone as a tendon. And so tendons attach muscles, many muscles to bone. So there's the gross anatomy. And if it's a tendon, it's called an indirect attachment. It's a tendon that attaches the muscle to the bone. A direct is that the epimysium merges with the periosteum of the bone, and there really isn't a tendon. Sometimes the tendons are sheets rather than cylindrical. And that's called an aponeurosis. A retinaculum is deep fascia that holds tendons in place. Right? Origins and insertions. This is important. So skeletal muscle, when the major, the role of skeletal muscle is move parts of your skeleton. Right? So the muscle is going to be attached to two parts of your skeleton, skeletal muscle. One part is called its origin. And so it's attached to one bone, and that's called its origin. And that part doesn't move. Then it goes to another bone where it inserts on that bone. And that bone moves. So your biceps inserts on top of your humerus right here. This is the origin. And then causes flexion of the arm like this. And its insertion is on the radius in the ulna, and when it contracts, it pulls up like this. So origin here, insertion here. Okay, so origin, part that doesn't move, the insertion, the part that does move. Okay. Muscle functional groups. Prime mover or agonist. This is going to be the main muscle that moves. If you look at a biceps, it's the main muscle that causes flexion of your forearm. There's one underneath called the brachialis that's a synergist. That's the next dash there. A synergist moves the bone in the same way that the prime mover does, but it's not as strong, and it's a smaller muscle. All right. Now, skeletal muscles exist in antagonistic pairs. As well, this is part of the joint lecture, which is extra credit. But I still have to introduce you to some movements. This is called flexion. This is extension. Right? So biceps causes flexion of your lower arm. Well, if there's a muscle that causes flexion, there has to be a muscle that causes extension, the opposite movement. So the biceps causes flexion, and there's a muscle back here, triceps, that causes extension. So if you move a muscle one way, you got to have another muscle on the other side to move the other way, right? So they exist in antagonistic pairs. Right? A fixator holds a bone in place. And sometimes this bone serves as an origin for other bones, and you want to keep it in place. So there's your shoulder blade is held in place in your back by muscles in your back that hold it in place, because it is an origin for a lot of these upper arm muscles have their origin, and the chest muscles have their origin, not the chest muscles, on the scapula, your shoulder blade. So there has to be other muscles to hold it in place. So, and you can actually move your scapula if you want. But if you move in your arm, you want to have that scapula, stationary, and that fixator holds it in place. Okay. Innervation. Skeletal muscle cells are innervated by a motor neuron, or a somatic motor neuron, whose axons make up spinal nerves or cranial nerves. So they come out, a nerve carrying axons of motor neurons comes out from the spinal cord, out the ventral root, through the spinal nerve, through a ventral or dorsal ramus out to the muscles, and they innervate the muscles. Blood flow, highly variable. If you're not using a muscle, your muscle just gets like a minimum of blood. But if you start using it, those blood vessels dilate, and you get a huge amount of muscle. So actually, we'll talk about this next semester in the cardiovascular system, how different organs get can get different amounts of blood if they're being used or not being used. And the most variable of all, actually the least variable is the brain. It gets a huge amount of blood no matter what. Maybe a little bit more, maybe a little bit less, but it's small. Skeletal muscle is the most variable. It gets a very tiny amount if it's not being used. It gets an enormous amount if it's being used. Okay, naming muscles. We're not going to get into naming muscles too much. Usually by how they move, like a flexor or an extensor, or this is called pronation and supination. Those muscles that do that are called pronators and supinators. Or about what part of the skeleton they move. They may mean names for that. But I'm not going to hold you to names of muscles. We're not going to do a lot of things about the names of muscles. Okay. So skeletal muscle, voluntary striated muscle. So it's voluntary. You have conscious control of it. Right? As a matter of fact, skeletal muscle cells are slaves to their motor neuron. They do not contract unless they're stimulated to by a motor neuron. This is called excitation-contraction coupler. How does the excitation of a muscle caused by a nerve, a motor neuron, lead to a contraction in the muscle? We're going to get to that later on in this chapter. So so it's striated. So it looks like you can see striations in a microscope. You can see striations, right? It has this in common with cardiac muscle. What it doesn't have in common with cardiac muscle is involuntary. You don't have control over your heartbeat. And then smooth muscle is called smooth because it's not striated. It's also involuntary as well. Okay. The cells in skeletal muscle are long, cylindrical, multinucleate cells called muscle fibers or myofibers. They're multinucleate. That's kind of an odd thing. We don't have too many multinucleate. We saw one multinucleate cell in the bone chapter, the osteoclast. Well, muscle fibers are multinucleate also because they're so long that in order to move, if you made a protein that had to be placed somewhere in the cell, and you only have one nucleus, this protein would have to be carried a very long way to be put in place. So since you have nuclei all along the way, you don't have to transport the proteins that far because any one of the nuclei can make this protein and put it in place right where the nucleus is. Okay. So as I mentioned, they're microscopically thin, but macroscopically long. They're from the fusion of myoblasts when you're an embryo. These myoblasts are undeveloped immature muscle cells, and they come together to form the myofibers. And that's pretty much it. Once you become an adult, you really don't make any new skeletal muscle. Right? There's a little bit of that, but very little. So if you go to the gym and you pump iron, you get big muscles, but you're not adding muscle cells to those muscles. You're just making bigger cells. Okay, this is called hypertrophy rather than hyperplasia. Hyperplasia is making new cells, and an organ getting bigger by making new cells. Hypertrophy is an organ getting or structure getting larger just by making larger cells. Okay. As I said, some myoblasts remain as satellite cells and can replace myofibers to a limited extent, but not that much. Generally, if you lose muscle cells, you're not getting them back. So it's like nerve cells in that regard, most nerve cells. So there's a myoblast fused to form a skeletal muscle fiber. All right, you can see multiple nuclei there. And there are some satellite cells left over that regenerate a little bit, but not too much. Okay. So we can talk about the muscle fiber, which is a muscle cell. The sarcolemma is the muscle cell membrane. The sarcoplasm is the cytoplasm in a skeletal muscle cell. Sarco is the prefix meaning muscle. All right, so you have glycogen stored in muscle. Remember, this is our stored carbohydrates. All right. Most of it's stored in what are called fast twitch muscles. We're going to get to that. That can contract really fast and go into anaerobic metabolism. But it's in all muscle fibers. And there's something called myoglobin in muscle also. It's a protein very similar to hemoglobin that carries oxygen in your red blood cells. So, but it exists in muscle, and it has even a stronger attachment to oxygen than hemoglobin does. So it's like a little oxygen tank in your muscles. So if you start really using your muscles, and the oxygen level gets really low, then the oxygen comes off the myoglobin, and you got a little bit of extra oxygen there. But that gets exhausted pretty fast after you start really moving muscles. Okay, the sarcoplasmic reticulum is really the endoplasmic reticulum of muscle cells, and it has a particular role as a storage area for calcium. And there are terminal cisternae that are right next to it that are part of the sarcoplasmic reticulum. There's transverse tubules, which are tubes attached to the sarcolemma that go down to the terminal cisternae and the sarcoplasmic reticulum, and so those three together are called triads. And this is going to be we'll talk more about this when we get into excentration and contraction coupling. How the action potential is generated on the surface of the sarcolemma and gets down deep within the muscle to open up calcium ion channels in the sarcoplasmic reticulum. That's going to lead to a muscle contraction. Okay. You have the myofilaments. All right. There's 3 myofilaments. The thick filament, which is really just myosin. Well, there's a enzyme in here called an ATPase also, but the thick filament is really just myosin. Thin filament has 3 proteins, main proteins actin. There's a long cylindrical protein called tropomyosin. And there's a group of small proteins collectively called troponin. And then there's an elastic filament called titin or connectin. And we'll see how these are arranged when we look at the internal anatomy of muscle cells. And we're going to look at how muscles actually work. It's called the sliding filament theory, where these thick filaments slide past these thin filaments. Well, actually, the thin filaments slide past the thick filaments. And so we're going to get to that a little later. Well, here's microscopic anatomy of a muscle cell. So here are these myofilaments, the thin and thick filaments in myofibril. And a sarcomere is, we'll get to sarcomere a little bit later. That's the smallest part of a muscle fiber that actually contracts. And these myofilaments are a series of sarcomeres, along with hundreds of sarcomeres, all in a linear array. So there's the sarcolemma, and here's a T-tubule in green that comes up from the sarcolemma down to the sarcoplasmic reticulum. And there are, and on the inside of the T-tubule is a terminal cisternae, which is part of the sarcoplasmic reticulum. And you can see here the sarcolemma and T-tubules that, remember, since muscle cells are excitable, they have voltage-gated sodium and potassium ion channels, just like nerves. And they also have sodium potassium pumps, just like nerves. And what happens is when the sarcolemma here is stimulated to threshold, and an exponential is generated, it travels over the sarcolemma and down into the T tubules. So along the sides of the T tubules are the same voltage gated ion channels. And so the exponential travels down deep within the muscle along the T tubules. And when it gets down there, it opens up calcium ion channels. And inside the sarcoplasmic reticulum, it's highly concentrated calcium. It's a calcium store, and there's almost no calcium in the sarcoplasm. So if you open up calcium ion channels in the sarcoplasmic reticulum, sodium will rush into the sarcoplasm, right? And that's going to bind to troponin, and we're going to initiate a contraction. So this is a little bit of excitation and contraction coupling right now. After the muscle relaxes, then calcium pumps are turned on in the sarcoplasmic reticulum. The ion channels are closed. Calcium pumps are turned on to resequester the calcium back into the sarcoplasmic reticulum, and that will cause muscle to relax. And so see, this whole cell is a bundle of myofibrils, right? Okay, so we got to know a lot of proteins that are in muscle cells, and contractile proteins and regulatory proteins. In the sarcoplasmic reticulum and sarcoplasmic reticulum membrane. So as you just mentioned, there are calcium channels and calcium pumps in the sarcoplasmic reticulum. Contractile proteins are myosin and actin, and they're the ones that like patch each other. Regulatory proteins are tropomyosin and troponin. Okay. Now, and we'll see how these are arranged in a moment. Sarcomere, arranged in sarcomeres. So there are bands in sarcomeres: I bands, H bands, A bands, Z disc, M lines. And these sarcomeres is what makes a skeletal muscle look striated. So here are two myofilaments. The thick filament on top, right here, and the thin filament down here. So thin filament is basically myosin. And a myosin molecule is like a double-headed golf club. It's got a tail here, and it's got a couple of heads right here with actin binding sites, and ATP and ATPase binding sites. And so all these tails are held together in this fiber, and the heads of the myosin are sticking up all the way around the myosin fiber. They're sticking out, right? Okay. Thin filament. The red balls are actin. So that's the main contractile protein in a thin filament. And there are myosin binding sites on every actin. But in a relaxed muscle, the myosin binding sites are blocked by this long fibrous protein called tropomyosin. And then every certain distance along the tropomyosin are the troponin complex. It's like three little proteins called troponin complex along the tropomyosin, and they have a calcium binding site on them. So, as I said, in a relaxed muscle, so we're going to have binding between myosin and actin in an active muscle, but they're not connected to each other in a relaxed muscle because the tropomyosin is blocking the myosin binding sites on the actin in a relaxed muscle, in a muscle that's not moving. Okay. So here's the arrangement of the sarcomeres. As I mentioned, a myofibril is a long series of sarcomeres. Here is the arrangement of the thin and thick filaments, plus the elastic filaments. So here are thick filaments like this, and in between them are thin filaments that are attached to a Z disc, right? A Z line. And then you have these bands. Now, an I band, on the microscope, an I band looks lighter than an A band because it only has these elastic filaments and the thin filaments, so it looks lighter. The A band has thin and thick filaments. Even though there's an H zone in the middle where it's only thick filaments, but they're thicker and it makes it look darker. And through most of the A band, we have both thin and thick filaments, so it looks darker under the microscope. So that's where you get these striations. Now, if you do cross sections, you'll see that particularly in the A band, every thick filament is surrounded by 6 thin filaments, right? And there are myosin heads that are pointing out to all 6 of those actins, all 6 of those thin filaments, okay? Okay, and when you get into I band, you see that there are connectin. Those are the elastic. These little springs are the elastic, and it may look like springs because they're elastic. When the sarcomere contracts. Now here's, like I said, the sarcomere is the smallest thing that contracts in a muscle. So what happens is the filaments slide by one another, and the Z discs come closer together. So it's contracted sarcomere. So sarcomeres contract when the muscle is stimulated. And each sarcomere contracts a microscopic amount, but every myofibril has so many sarcomeres in a row that all these sarcomeres contracting a microscopic amount adds up to a big macroscopic contraction. Okay, and then when a sarcomere is contracted, these elastic things are like stretched rubber bands. So then when you stop when you turn the muscle off, then the Z discs are pulled apart, and then the elastics pull the Z discs apart in a relaxed muscle. Okay. So like I said, the connectin or titin is the elasticity there from the elastic fibers. There's something called nebulin, which is a protein that orders the structure of the sarcomere. There's something called dystrophin, which anchors biofilaments to the sarcolemma. I'm not sure what this thing does, but there's a genetic disease called muscular dystrophy that leads to really seriously impaired muscle functioning, and this is a genetic disease. It's a mutation that has a dystrophin not working properly, and somehow that leads to really impaired muscle function. I don't know much about that. Okay. So mitochondria and other structures. Obviously, muscle cells have mitochondria. Many mitochondria per muscle cell. And it depends upon the type of fiber we're talking about. Now, this is, later on in the chapter, we'll get into what are called fast twitch fibers and slow twitch fibers. And fast twitch, when we get into the metabolism, muscle cell metabolism, fast twitch fibers generally use anaerobic respiration. And that means they don't use mitochondria. They have mitochondria, but they don't have that many. Then you have the slow twitch muscles that have lots and lots of mitochondria, and they generally don't use anaerobic metabolism. They use aerobic metabolism. Because remember, oxygen is used in mitochondria. Okay, I already mentioned the myoglobin out there. There's also something called creatine phosphate, which is a really quick source of ATP. ATP cannot be stored. It's much too unstable a molecule. It's only made as it's used. Right? But there are certain sources of ATP that can be faster or slower. An anaerobic source of ATP is quicker than the aerobic source of ATP. And creatine phosphate is even quicker. It's almost like stored ATP. So it's a really quick form of ATP. So you can move your muscles really quickly. Okay, nerve-muscle relationship, motor neurons and motor units. Axons of motor neurons called somatic motor fibers. A motor unit is all of the muscle fibers innervated by the same motor neuron. So a motor neuron innervates, has axon collaterals and can innervate more than one muscle fiber. The size is variable. How many muscle cells are innervated by this particular motor neuron? It's small in very small, the motor unit is small, that is, there's only a few motor neurons, few muscle cells innervated by a motor neuron in small muscles that do fine work, like extrinsic eye muscles and muscles that control your fingers. Large in calf muscles and back muscles, the motor units are quite large. Could be a couple of hundred muscle cells innervated by one motor neuron. Where in your fingers, maybe there's only 10 or a dozen muscle cells innervated by the same motor neuron. And remember, muscle cells are slaves to the motor neuron. If the motor neuron sends an action potential to them, they contract. It's always excitatory. The muscle synapses are always excitatory. All right. Okay, and they work in shifts. If you hold the upset in a long time, you have a certain number of motor units that are firing, and they get fatigued. And so those, and this is like an unconscious thing. So there are some things that work in skeletal muscle that are unconscious, and that is shifting to different motor units. If you keep on holding this heavy thing, then some motor units will get fatigued, they'll be turned off, and other motor units will be turned on to keep on holding this up. If you make it heavier, then you have to fire more motor units to hold up something heavier. So the tension that is generated by a muscle is determined by how many motor units are fired. Or the neuromuscular junction, or the motor end plate. All neuromuscular junctions use acetylcholine and are all excitatory. So if you want to inhibit a muscle from firing, you have to inhibit the motor neuron. You don't inhibit the muscle, you inhibit the motor neuron. And motor neurons always excite skeletal muscle fibers. Okay, the motor end plate is what's called the postsynaptic membrane. So when we look at synapse, the neuromuscular junction is a synapse, and it's exactly the same as all the synapses we talked about in the nervous system chapter. It's the same. It's the same parts. It's cholinergic. And the presynaptic membrane is the motor neuron axon terminal, and the postsynaptic membrane, called the motor end plate, is a sarcolemma. That's the muscle cell membrane. So here's a couple of motor units here. So it's two motor neurons, one in red, one in blue. And so the red ones innervate the red muscle fibers, the blue ones innervate the blue muscle fibers. And so if the red motor neuron is stimulated, it will cause a contraction of the red fibers, not the contraction of the blue ones. Even though the fibers are right next to each other, that endomysium electrically isolates every muscle cell from every other muscle cell. So even though two muscle cells might be right next to each other, if they're innervated by different motor units, if one is contracted, the other one will not be contracted. So in order to contract the blue ones, they're in a different motor unit, you have to stimulate the blue motor neuron. And there's a neuromuscular junction, excitation of the skeletal muscle fiber. It's, as I said, it's exactly the same as the synapses we learned about in the nervous system. So here's a nerve signal coming along the synaptic knob. It opens up voltage-gated calcium channels. Calcium enters the synaptic knob. It causes the vesicles containing neurotransmitter to merge with the presynaptic membrane and exocytose the acetylcholine into the synaptic cleft. It goes across, diffuses across the cleft, binds to acetylcholine receptors on the motor end plate, and this opens up sodium ion channels, and the postsynaptic membrane, sarcolemma, has a motor end plate potential, a gradient potential that will spread out into the regular sarcolemma, and right on the surrounding the synapse, the sarcolemma surrounding the synapse has voltage-gated sodium and potassium channels, and that surrounding. So if the synapse fires, that surrounding sarcolemma will be brought to threshold, and an action potential will be generated in that muscle cell and go out in both directions. So release of acetylcholine from the synaptic knob, binding of acetylcholine to acetylcholine receptors, and this causes a motor end plate potential, that gradient potential. Okay, skeletal muscle cells are identical to neurons in that they're capable of generating and conducting action potentials in the very same way. Excitation, muscle cell resting membrane potential is more negative than neurons. It's minus 90 millivolts. And that gradient potential, but a firing of synapse, as I mentioned, is called a motor end plate or an end plate potential. So this will lead us into what's called excitation, contraction, coupling. How the excitation of the sarcolemma leads to a muscular contraction. And we're going to be, I'm going to end it there and pick up on excitation, contraction, coupling in the next video. Okay, we'll stop here.